This Embryology Highlights screencast, Head Development 1, presents the development of the pharynx and pharyngeal arches. The pharynx is the rostral end of the foregut, which is formed during development of the head process and lateral body folds. The pharynx is closed by an oropharyngeal membrane formed by endoderm on the inside and ectoderm on the outside. The membrane disintegrates when a stomadeum grows outward from the ectodermal surface. The pharynx is bounded laterally by pharyngeal arches. Internally, the pharyngeal wall has expansions called pharyngeal pouches. Both arches and pouches develop into various adult structures. On the left, we have a surface view of an embryo. 3 pharyngeal arches are visible in mammals. The forehead and nasal cavity will be formed by this blue-colored area, which is a frontonasal prominence. On the right, we have a mid-sagittal section through an embryo. Notice that the pharynx ends blindly in an oropharyngeal membrane that eventually disintegrates when a stomadeum grows out of the cranium. This image represents a dorsal plane section through the pharynx. We see the floor of the pharynx, and we see pharyngeal arches that have been transected. Pharyngeal arches are demarcated by external pharyngeal clefts and by internal pharyngeal pouches. In fish, pharyngeal arches are called branchial arches. Branchial means gills. In fish, the tissue separating these branchial arches degenerates so that water can flow from the pharynx to the exterior, and these arches then become gills. This image shows surface views of 3 stages of embryonic development. 3 pharyngeal arches are evident in mammalian embryos. The first arch, shown in yellow, develops maxillary and mandibular processes which grow out and become upper and lower jaws. The second arch forms hyoid bones. The third arch contributes to the tongue. The fourth and sixth arches merge. The individual arches are separated by endodermal pouches and ectodermal clefts. Each arch contains ectomesenchyme, derived from neural crest, an aortic arch artery, myotomes that come from semitomeres, and a cranial nerve that innervates the arch and whatever it becomes. Semitomeres, which develop anterior to the notochord, are less developed than somites. They give rise to myotomes that migrate into pharyngeal arches and develop into muscles innervated by the cranial nerve that innervated that arch. Pharyngeal arches contribute to a number of head structures. In addition to upper and lower jaws, pharyngeal arches contribute to the tongue and the larynx. The ectomesenchyme found within pharyngeal arches and within the frontonasal prominence gives rise to intramembranous bone of the face and calvaria. Many skeletal muscles of the head are derived from pharyngeal arch myotomes, which come from semitomeres. Each pharyngeal arch receives one cranial nerve, which innervates all of the muscles that are derived from the myotomes that go to that arch. The first pharyngeal arch gives rise to the muscles which move the jaws, the temporomandibular joint. The second arch gives rise to muscles of facial expression. The third pharyngeal arch gives rise to one muscle of the pharynx, and the combined fourth and sixth arches supply muscles of the pharynx, larynx, and esophagus, those skeletal muscles. This image illustrates myotome migration to pharyngeal arches. The light pink area shows the beginning locations of somite and semitomere myotomes. The dark red patches show where myotomes migrated to pharyngeal arches and to the orbit and to the tongue. Each pharyngeal arch receives a cranial nerve which innervates the myotomes that migrated to that particular arch. This image shows the face and the cranium of a dog skull. The The bones of the upper jaw and those of the calvaria develop intramembranously from ectomesenchyme that is located within the first pharyngeal arch and within the frontal nasal prominence. Bones at the base of the cranium are derived from sclerotomes, and they develop endochondrally. Mandibular development is more complicated, involving both endochondral and intramembranous phases of development. This image illustrates pharyngeal arch contributions to tongue development. On the right, we see the tongue color-coded according to developmental regions. The root of the tongue develops from a proximal swelling and also from contributions from the third pharyngeal arch. The body and apex of the tongue develops from paired distal swellings and from first pharyngeal arch contributions. The second pharyngeal arch contributes taste innovation to the tongue. The larynx, which develops from pharyngeal swellings, gets contributions from the combined fourth and sixth pharyngeal arches. Pharyngeal pouch endoderm gives rise to various cavities and glands of the head. The first pharyngeal pouch gives rise to the auditory tube and middle ear. The second pouch gives rise to the fossa for the palatine tonsil. Finally, the thyroid gland develops from a midline evagination from the floor of the pharynx. We're looking at a surface ventral view of the pharynx, and these are the various pouches, and this coming from the ventral midline is the thyroid gland. This image shows a dorsal plane section through the left wall of the pharynx at early and later stages of development. The first and second pharyngeal arches are separated by the first pharyngeal pouch and the first pharyngeal cleft. The first pharyngeal pouch becomes the auditory tube and the middle ear. The first pharyngeal cleft becomes the external auditory canal. And the intervening endoderm-ectoderm tissue becomes the tympanic membrane, in other words, the eardrum. The second pharyngeal pouch becomes the fossa of the palatine tonsil. Remaining pouches become glands and thymus. This textbook image shows species variation regarding the thyroid gland. Since the thyroid gland develops from a single midline evagination of the pharynx, there is only a single thyroid gland, which is what you see in the pig. Here's a lateral view and a transverse view showing the trachea and the single thyroid gland. However, the canine thyroid gland has left and right lobes, and in cattle and horses, the left and right lobes are connected by an isthmus. This concludes this embryology highlight screencast concerning head development of the pharynx and pharyngeal arches.